Hello everyone, this is Sean Taylor, Field Application Scientist Manager for GenScript in North America. And as many of us work from home with our families to blunt the rate of infection globally for COVID-19, there are multiple hospital, university, pharmaceutical, and government research institutes that are working very hard towards a treatment for this pandemic. Here we'll explain some of the pathways for treating and eradicating coronavirus with solutions to minimize time to results. So, first we'll discuss the virus structure, then the mechanism of infection. These are both important to understand how we can treat the disease. If we don't understand the virus itself, it's hard to determine a pathway towards efficient treatment. Potential treatments currently being explored and the requirement for purified antibodies and proteins for these treatments, and then followed by the tools that are used to actually perform this protein or antibody purification. The two main ones that are available are column-based protein purification, and there's a new emerging technology, magnetic bead-based protein purification. So I'm briefly going to talk about the virus only to bring in how we can treat the disease. The structure and nomenclature, this section of the presentation has already been described in this YouTube video. So if you go to this YouTube video, you'll get all the details. The only thing that I really want to go into a little bit of detail here is on nomenclature. It's just important for when you're looking at the literature or when you're looking at the television or talking to people, that some people will use interchangeably COVID-19, SARS-CoV-2, or 2019 nCoV to delineate the virus, and these all mean the same thing. It's also important to understand, and this is important for treatment, that it's this spike glycoprotein that is a real key to the way this virus infects people. So the spike glycoprotein binds to the angiotensin converting enzyme 2, the ACE2 receptor in the epithelial cells of the deep lung. It's also important to understand that there's RNA inside the virus and that this RNA finds its way into the host cells, into the epithelial cells, to replicate the virus. So when we talk about transmission mechanism of COVID-19, I've already kind of elucidated that in that it's through this spike glycoprotein. So this globular structure here would be the virus. And here's a spike glycoprotein, there's another one, and there's another one. And this particular spike glycoprotein is binding to the ACE2 receptor of an epithelial cell. So this here would be an epithelial cell in the lining of the lung. And you can see now the virus is tightly bound. And when it's bound, it will internalize into the cell and then release this RNA that's inside the virus to generate more virus particles. And what you see here is a viral particle emerging from the cell. The cell will burst eventually because it'll be filled with virus, and more virus will be replicated and made to infect more and more cells in the lining of the lung, and this leads to the symptoms of disease. So by understanding how the virus recognizes the cells through this ACE2 receptor and the spike glycoprotein, we can start to consider how we can treat this disease. So the first level of treatment, obviously the cure, the ultimate treatment, is a vaccine. And there are people working on vaccines that are not just to the proteins that interact with the epithelial cells in the virus, but also even to the RNA. So some vaccines are being made against the RNA inside the virus. So this is interesting and hopefully will lead to a very nice viable treatment for coronavirus down the road. But these treatments take a while, months to even over a year. So we're still trying to come up with a midterm solution, and that's where these shorter treatment options come into play. So for shorter treatment options, and what's actually worked successfully in the past, is the purification of antibodies from the serum of COVID-19 recovered patients. So in the past with other viruses, this has been performed successfully where you take the serum from a recovered patient and the recovered patient will have generated an immune response to the virus and therefore will have circulating antibodies to the virus, and you can purify those antibodies and then re-inject them into a patient who's suffering from disease, and hopefully those antibodies will attach themselves to the virus and prevent infection. So this is an option, and this is described here in this paper. But however, with a global pandemic at the scale that we're at, with so many thousands of people infected and in serious condition, it's not a viable option to go and take blood from thousands of people to use them to help with curing disease in infected people. So we're looking for a more sustainable option, a more scalable option than this one. So the next option could be the screening and production of neutralizing antibodies to COVID-19. So again, this is described here, and it's been previously explored for SARS-CoV, Nipah, and Hendra viruses, and described in this article here as well. So these are options too, where you can make neutralizing antibodies that bind all around the viral coat here. And by having antibodies bound all around the virus, they'll block the interaction between the spike glycoprotein and the ACE2 receptor, preventing the virus from binding to the cell and therefore internalizing into the cell and forming more viral particles. So essentially you're blocking the mechanism of infection of the virus. And the third option would be to characterize the receptor binding domain of COVID-19. So the receptor binding domain, as I explained, is here. So we have the S glycoprotein, so the spike glycoprotein. The receptor binding domain is the S1 domain, which is in yellow here. And if we can characterize this really well, which has been done, the crystal structure of this has been already elucidated, then we can start to come up with novel drugs, chemicals, or proteins or antibodies that might bind here to the spike glycoprotein and block it from binding to the ACE2 receptor. So therefore you've generated a chemical that blocks the interaction. So all of these treatments are viable options, and this has been described in this article here. But all of these treatments, vaccine development, purification of antibodies, production of neutralizing antibodies, and the production of proteins or antibody partners that might block the interaction between COVID-19 and the ACE2 receptor, all require purification. Many of the potential treatments for COVID-19 will likely require purified proteins, and column-based chromatography is primarily used for protein purification within the scientific community. But it does have some drawbacks. So here is an image of a typical chromatography system where we have an instrument that pumps a liquid sample onto a column, through which the protein of interest will gradually separate away from the other proteins in the sample, eluting to the purified product. Now, when treating a disease with purified protein, we need to test hundreds or thousands of different proteins for their efficacy in treating disease. So the ideal purification system would therefore have a very high throughput. Now, unfortunately, with column-based purification systems, the throughput of a system comes at a price. So the cost goes up when throughput goes up. Furthermore, these systems are fairly complex to use, both in the setup of the system and in the programming of these systems. So the reality is, if you want throughput, you have to pay for it, and you also need to be trained for it over a period of time. And that learning curve can be weeks to months, time we don't have in a pandemic situation. Also, these high throughput systems don't permit the loading of large volumes, so that they have limited sample volumes. Milliliter quantities of samples can be loaded on these systems, and low milligram quantities of protein are typically eluted, which may not be enough to test those proteins in a number of different treatment options. Also, the harsh reality of these systems is that they are not as high throughput as potentially made to believe, because there is costly and timely sample preparation required for these systems. The reason why the sample prep is because many protein samples contain either cellular debris or protein aggregates that can clog the column. So if the sample is not cleared of those aggregates and cellular debris, then the protein will never be able to be loaded on the column because the column will be clogged and it'll block the protein from being separated. So that requires centrifugation and filtration of the sample. Centrifugation and filtration require a large amount of time. So therefore, the throughput that we thought we were gaining is reduced. We have less throughput because we have to take more time to prepare the sample by centrifugation and filtration. And that's the harsh reality that a lot of manufacturers of these high throughput systems don't discuss, the upfront preparation. Also, we see that this bubble is a little bit higher in cost, and that's because of the filtration. So not only does filtration add time, it also adds cost. We also have another option for purification, magnetic bead-based purification. And this is a great option because it's much more scalable and it's much less complex than using a column-based system. So magnetic beads have been around for a while, and Amgen and GenScript worked together to pursue a new application, which was to actually purify large amounts of protein with magnetic beads. And it's as simple as just adding the magnetic beads into a live cell culture, where the protein's being secreted, obviously. And then the washes and elution steps can all be performed then with a magnet to give purified protein. So in its essence, magnetic bead-based purification doesn't even require a system to do this. This can all be done manually and very quickly. So if we look at the concept itself in a little more detail, we see cell cultures here, and these cell cultures are secreting protein into the medium. So these would be the proteins that could potentially be used to treat disease, either antibodies or proteins. And then the beads are added directly into the cell culture, typically either the night before or the morning of harvesting the cells. They don't affect the growth of the cells, so they can be added directly into the cell culture, and that's why this cell culture looks darker, because the beads themselves are a darkish color. And then just by incubating in the culture for several hours or overnight, you can then purify, go from sample to flow through to purified protein with no centrifugation or filtration or no extra loading time. So we're avoiding a bunch of steps that increase the complexity and the cost and the time of getting to the final protein. And especially in the current situation with a global pandemic where we're trying to reach our final treatment as fast as possible, we want to be able to do all of these research steps as quickly as possible so we can test these purified proteins as a treatment. So with mag bead-based purification, there's no clarifying steps needed. So we go straight from mag beads to incubation in the sample, to washing and elution without any clarification step. As I mentioned, we add the beads directly in the cell culture. There's no need to centrifuge or filter. However, with resin-based purification, and that gets back to column-based systems, there's definitely a requirement to centrifuge, and there's often, in many cases, the requirement to filter samples before we load them on those column-based systems. Because if we don't do these two steps, the column will clog, those debris will clog the column and prevent the protein from flowing through the column, and therefore getting our purified protein, which we need for treatment, as fast as possible. So there are a variety of methods for manual purification that we see here. So we have, for smaller volumes, 96 well plate magnets if you're looking at microliter quantities. If you're looking at milliliter quantities, there are 15 and 50 mil conical tube racks that can be used for 15 to 50 mils. And then we have a magnetic block and a magnetic rod for looking at larger volumes. So this would be for liter quantities of protein samples. All of these are very simple, easy to use to just perform protein purification manually in the lab in a matter of minutes. So for the rod, so this is more important when you're looking at larger culture volumes. You have your beads that are swirling in the solution. So these beads are incubating with the sample and drawing the protein to the beads. You add the magnetic rod in, and the rod scoops up, literally locks the beads onto this magnet. So you see there's no more beads left in the culture. They're all bound to the rod, and those beads are bound to your protein. So now the rod can be transferred into a smaller vessel to perform the washes and elution steps. Same thing with the magnetic block. It's the same principle, except the magnetic block magnetizes the beads to the bottom of the culture flask, so you can pour off the residual cell culture to then transfer the beads into a smaller vesicle for protein purification. And all of this is very quick and easy to do with the magnetic bead-based technology. There is a semi-automated system, so if there is a need to go faster, to reach those purified proteins quicker so they can be screened and assessed for treatment options for COVID-19, then there is the AMMAG assay. And this system permits the automated purification of up to 12 samples at a time. So it's very convenient, permits the purification of 12 samples in a matter of about 40 minutes. So this permits the purification of up to about between 1 and 72 samples per day by using this system. And what this system will do is all the washes and elution steps simultaneously for 12 samples. So all the manual washing and elution that would be done by hand is done by this system for 12 samples in about 40 minutes. So it's very convenient. Protein can be purified very quickly, 80 milligrams per sample, and for very small volumes of samples. So it's very convenient to work with. And the operating cost, again, is very low, because if these beads are purchased in bulk quantities, it's a very low dispensation cost to purchase beads and then to use them to purify proteins. There's a large number of beads available in this case. So we have protein A beads, G beads, and AG and A beads. So whatever flavor of antibodies you happen to be purifying, likely the material is available to help you do this quickly and efficiently. So this is the goal here when we're trying to get proteins into the pipeline to test for treatment options. The ones we recommend typically for antibody purification are the protein A AMMAG beads, because they have a very high binding capacity, greater than 40 mg per mil, and they're alkaline tolerant, so it permits the beads to be incubated with up to 0.1 normal NaOH to eliminate endotoxins, and also to permit them to be reused several times with culture after culture, up to about 40 times. So it makes it very convenient, and again reduces the operating cost dramatically. So if we compare the purity and recovery of resin versus the magnetic bead technology, we can see that for multiple samples, the recovery was very, very similar, going all the way across through all of the multiple samples, maybe even a little bit better with the AMMAG, with the magnetic bead technology. On the purity side, again, we tested the same 4 samples with resin and with magnetic beads, and we see that the purity of the antibodies from those 4 samples with resin were identical to the magnetic beads. If you're purifying tagged proteins for downstream work to produce kits or treatment options for COVID-19, there are a large number of tagged resins, nickel, glutathione, streptavidin, and flag tagged resins that are all available with various binding capacities, various costs depending on what you're doing and depending on the throughput you have, to be able, again, to quickly purify protein. As with the antibody beads, the protein A and G beads, these beads can be incubated directly in cell culture or in a crude lysate to be able to purify protein. So what we have here is the sample, the flow through, and the purified protein from a resin, from a column, after centrifugation. We have the same 3 samples post-centrifugation for magnetic beads. But what we also tried was the same 3 samples without centrifugation. So these are the beads added into the crude lysate to just look at the differential data, and the resulting purified protein without centrifugation or with centrifugation was pretty much identical. So these beads can be added directly into a crude lysate, so there's no need for centrifugation or filtration for purifying protein targets or antibody targets. So again, the purpose here is to reduce complexity, to make things go faster, make it easier, reduce the need for complex equipment in the lab and complex training and a steep learning curve, which would take more time to ultimately lead to purified protein, which is what's needed to treat disease. There's another technology that we have available as well for the downstream. So again, the goal here is to move things as fast as possible. And when you're purifying proteins, you're often running gels, Kumasi stained gels, to be able to visualize the proteins after purification. So these bands are proteins, we're trying to visualize them. We run a gel and we stain with Kumasi. This is another bottleneck in the protein purification process. And we have an instrument called the E-stain, which eliminates that bottleneck. So each of these units permits the staining and destaining of up to 2 gels in approximately 9 minutes. So it goes very fast, much easier and quicker to reach the final data that's needed to know if these proteins are pure. So GenScript not only offers a novel and practical protein purification solution to help accelerate treatment of COVID-19, we also have a number of purified proteins to the virus and antibodies and ELISA kits for early detection of COVID-19. All of these products and services are assembled here on our website. So if you click on this link, it'll take you to the website where you can peruse the early detection assays. So we have a qPCR assay, ELISA kits, and proteins and antibodies to assemble custom kits, as well as services to help with producing novel proteins or antibodies to help in the production of early detection assays. Furthermore, we've recorded a video for early detection of COVID-19 if you wish to learn more. And finally, if you click on Catalog Products under Protein Isolation and Purification, you can access our magnetic bead-based technology and automated magnetic systems, as well as for manual production of purified proteins. To conclude, please continue to practice social distancing, stay safe, as we work through with the research community to come up with fast solutions for both treatment and early detection of COVID-19.